Dear all, welcome to stem cells and tissue engineering. Today we are going to see introduction to the stem cells. Good morning, one and all. This is Selvakumar, Associate Professor, Department of Agricultural Biotechnology, Bharat University. I'm happy to welcome you all to my class, the introduction to the stem cell engineering. And today we are going to see stem cells. Very, very interesting topic. So before starting my class, we should know what is cell and what is stem cell. So what is cell? Cell, it may be a plant cell, it may be animal cell or microbial cell. What is cell? The definition of cell is the basic structural, functional, biological unit of all living system is called cell. Basically, the word cell derived from the Latin word cella. It means that the small room. So cell is the structural, functional unit of all living system. So what is stem cell? Stem cell is a normal cell, and those cells that have the capability of self-renewal and differentiation are known as stem cells. So self-renewal is the ability of cells and to proliferate without a loss of a differentiation potential and without undergoing the senescence. A stem cell is like a blank cell, that is precursor cells, that can give rise to multiple tissue types such as skin cells, muscle cells, or the nervous cells. And the stem cell is very essential to building block of the human body. Right. The stem cells have a different type of features. What are the essential features of the stem cells? Stem cells are very unique cells. And second point, the stem cells have the amazing ability to develop and distinct cell types in the human body. Third point, the stem cells can be used as a repair system for the human body. And fourth important characteristic feature of the stem cell is the stem cells can theoretically divide without any limit in living organisms in order to replenish the various type of cells. The important fifth character, when a stem cell divides, each new cell has potential to either remain a stem cell or become another type of cell with a more specialized functions. For example, red blood cells, muscle cells, nervous cells, etc. Next, I'm coming to, there are three unique properties of the cell, stem cells. There are three unique properties of the stem cells. What are the three important properties of the stem cells? The stem cells are capable of dividing and renewing for longer periods. Second point, they are unspecialized and they can give rise to any specialized cell types. A stem cell is uncommitted cells until it receives a signal to develop into a specialized cells. Next, I'm coming to the unique features of the stem cells. Already we are discussing about the what is stem cell and the unique features of the stem cell. Now the points may be extended, and a stem cell is an unspecialized cells that develops into variety of tissue types or organ types, especially the specialized cell types. A stem cell divides and give rise to give rise to one among the cells and the additional stem cells and a specialized cells. For example, I can say a hematopoietic stem cells. The hematopoietic stem cell produce a second generation of a stem cell and a neuron. And a progenitor cells, that is precursor cells, unspecialized, and that is capable of undergoing the cell divisions and yielding the two specialized cells. For example, the myeloid progenitor or the myeloid precursor cells, the undergoing cell division yield two specific specialized cells, that is neutrophil and red blood cells, neutrophils and RBC, that is red blood cells. Next, I'm coming to the classification of the stem cells. And the classification of the stem cell basically based on the potency of the stem cells. So what is potency? Potency is nothing but potential, the ability of the cells. So the potency or the potential of the stem cells defined by the types of differentiated cells, that is stem cells, that the stem cells can make, and either number one, totipotent, pluripotent, multipotent, and unipotent. What is totipotent? Toti means totally potential. Pluri means partially potential. Multi means somewhat potential. Unipotent, that means somewhat little bit potential. So totipotential, partially potential, somewhat potential. That is totipotency, pluripotency, multipotency, and unipotency. Now let us see one by one. What is the potential of the stem cells? That is totipotential. I tell you, toti means totally. Potent means potential. Toti, potential, that is totally potential. And these cells have the capability to produce all types of the developing organisms, including both embryonic and extra embryonic tissues. For example, placenta. So, totipotential cell can give rise to the complete organism. It may be a human being, maybe animal, or whatever it may. So, totipotentials have the ability, it can give rise to a complete organism. It's called totipotential. Next, I'm coming to pluripotential. What is pluripotential? Partially potential. So partially potential cells can only make the cells of embryo properly, but make all cells of the embryo, including the germ cells and the cells from any of the germ layers. Therefore, they can make any cells of the body. It can make any cells of body, or it can give rise to any tissue types or any organ types, right? Partially potential cells. Next, I'm coming to multipotential cells or multipotent cells. Multipotent cells can only make the cells within a given germ layer. For example, the stem cells from a mesodermal tissues like blood, it can make all the cells of the blood, but it cannot make cells of the different germ layer, such as neuronal cells, ectodermal origin, or liver cells for the endodermal origin. So multipotent cells can give rise to a part of cells which is a part of organs, etc. And unipotent cells. The cells make cells of a single cell type. For example, a germline cell and a stem cell that makes the cells that mature to become egg or the sperm, but not all type of cells. Next, I'm coming to the embryonic type of stem cells. What do you mean by embryonic type of stem cell? It is embryonic stem cells. The embryonic stem cells are EES cells, mostly pluripotent stem cells derived from the inner cell mass of a blastocyst. Inner cell mass of the blastocyst. It's the early stage of pre-implantation embryo. Pre-implantation embryo. What is blastocyst? The next question arises. When a single sperm reaches to a single egg, the fertilization takes place. It is zygote. Zygote basically a single cell. Then after the cell division, the zygote becomes 2 cells, then 4 cells, 8 cells, 16 cells, 32, 64, 128, 256. It multiplies. So once a zygote formed, the 5th day or 6th day cells are known as blastocysts. It looks like a ball-like structure. So inside the ball is called inner cell mass. The outer membrane of the ball is called outer layer. Mostly totipotential cells are derived from the outer membrane of the blastocyst. And pluripotent cells are inner cell mass. That is inside the ball-like structure. No? It is inside. It is there. So it is called inner cell mass. Most of the cells are derived from inner cells. So inner cells are the pluripotent stem cells. Next, I'm coming to the embryonic stem cells. What are the characteristic feature of embryonic stem cells or ES cells? The human embryos reach the blastocyst stage, that is 4th day, 5th day, and 6th days, the post fertilization, at which time they consist to 50 to 156. Remember, 50 to 156. Isolating the embryoblast, that is inner cell mass, or we call it as ICM, inner cell mass, it results in the destruction of the blastocyst, which raises the ethical issues, including whether or not the embryos at the pre-implantation stage should be considered to have the legal status as more developed human beings. So most of the countries, this kind of experiments is prohibited. Next, I'm coming to in continuation of the embryonic stem cells. The embryonic stem cells of the inner cell mass are pluripotent and able to differentiate to generate the primitive ectoderm, which ultimately differentiates during the gastrulation into all the derivatives of the 3 primary germ layers such as ectoderm, endoderm, and mesoderm. Finally, these include each of more than 220 different cell types in the adult human body. How many cells? 220 different cell types in the adult human body. Right. Next, I'm coming to the pluripotency of the embryonic stem cells. So the pluripotency of the embryonic stem cells distinguishes the embryonic stem cells from the adult, while the embryonic stem cells can generate all cell types in the human body, especially the adult stem cells are multipotent and can produce only a limited number of cell types. If the pluripotent differentiation potential of the embryonic stem cells could be harnessed in vitro, it might be a means of deriving cell or tissues virtually to the organ. Next, I'm coming to in continuation of the embryonic stem cells. Additionally, under the defined conditions, the embryonic stem cells are propagating themselves and indefinitely in an undifferentiated state and have the capacity when provided with the appropriate signals to differentiate, presumably through the formation of the precursor cells to almost all mature cell phenotypes, this allows the embryonic stem cells to be employed as the useful tools for both research and regenerative medicine, because they can limitless number of themselves for continued research or the clinical purpose. Sir, one word newly you are pronounced, regenerative medicine. What is it? The next question arises. Basically, regenerative medicine or the reparative medicine is nothing but stem cell based therapeutics. Stem cell based therapies. The stem cells can be used to treat various genetic disorders. That is called regenerative medicine or reparative medicine or stem cell based therapies. So is it possible to cure a cancer with the help of stem cells? Yes, absolutely. So stem cells has the ability to cure or to treat many different type of human illness, diseases or disorders. How? We'll see one by one. Next, I'm coming to the advantages of the embryonic stem cells. Remember, I told you the ESC, that is embryonic stem cells, is one of the vital tool to treat many types of human diseases. What are the basic advantages of the embryonic stem cells? Number one, the flexible appears to have the potential to make any cells. Number two, it's immortal in nature. One embryonic stem cell line can potentially provide an endless supply of the cells with the defined characteristics. Very important point. The availability of embryos from in vitro fertilization clinics nowadays, right? Infertility problems are most vital problems for male or female. So this technique availability, this embryonic stem cells availability embryos from the in vitro fertilization clinics or the IVF clinics. Next, I'm coming to the limitations of embryonic stem cells. What are the limitations or drawbacks of the embryonic stem cells or disadvantages of the embryonic? I can give you 4 to 5 points regarding the limitations of the embryonic stem cells. Number one, difficult to differentiate uniformly. So underline, difficult to differentiate uniformly and homogeneously into the target tissues. Number two, immunogenic emission. What is immunogenic? The immunogenic embryonic stem cells from a random embryo donor are likely to be rejected after the transplantations. And third point, tumorogenic. So these embryonic stem cells are capable of forming tumors or promoting the tumor formations to human body. And number four, the destruction of developing human diseases. It may destroy the human life. Next, I'm coming to very important topic, adult stem cells. Remember, I told you totipotent, pluripotent, multipotent, and unipotent. Basically, adult stem cells are multipotent stem cells. So, what is the definition of the adult stem cells? How will you define the adult stem cells? And the adult stem cells is thought to be an undifferentiated cells among the differentiated cells in a tissue or organ is called adult stem cells. And the adult stem cells can renew itself and can differentiate to yield some or all the major specialized cell types of the tissues or organs. Unlike the embryonic stem cells, the adult stem cells are defined by their origin. The cells from the pre-implantation stage embryo. The origin of adult stem cells in some mature tissues is still under investigation. So next, the primary role of the adult stem cell in a living organisms is to maintain and repair the tissues in which they are formed. And scientists also use the term somatic stem cells instead of the term adult stem cell, where the somatic refers to the cells of the body and not the germ cells or sperms or eggs. So, how the differentiation pathways of adult stem cells? So, the differentiation pathways of adult stem cells. First, the classical example is the neural stem cells. So, how will you define the neural stem cells, or what are the characteristic features of the neural stem cells? In the brain, it can give rise to the three major cell types. Number one, the nervous cells or glial cells or neurons. And the second category, the non-neuronal cell types such as astrocytes and oligodendrocytes. The third category of the cells, the epithelial stem cells in the lining of the digestive tract, occurs in deep crypt and give rise to the several cell types. For example, absorptive cells, goblet cells, panic cells, and endocrine cells. Next, I'm coming to the differentiation of pathways. And the second classical example is the skin stem cells. And basically occurs in the basal layer of the epidermis and at the base of hair follicles. And the epidermal stem cells can give rise to the keratinocytes, which migrate to the surface of the skin and form a protective layer. So the follicular stem cells can give rise to both the hair follicles and to the epidermis. Next, I'm coming to the advantages of adult stem cells. Adult stem cells from the bone marrow and umbilical cord appears as flexible as the embryonic type. And second important thing, somewhat specialized, inducement may be simpler. It's not immunogenic. The recipients who receive the products of their own stem cells will not experience the immunological rejections. There are 4 different types of immunological rejections. So it's not tumorigenic. That means it tends not to form any tumor to the human being, and it never harm to the donors. Next, I'm coming to the disadvantages of the adult stem cells. Number 1, the limited quantity it can sometimes be difficult to obtain in the numbers. Number two, the finite may not lie as long as the embryonic stem cells in culture. It means finite. Finite means what? It may not live as long as the embryonic stem cells in the culture medium. Third important point, less flexibility, and it may be more difficult to reprogram to form other tissue types. These 3 points are the disadvantages of the adult stem cells. Limited quantity, finite, and less flexibility. So next I'm coming to IPS. So what is IPS, sir? Induced pluripotent stem cells. Induced pluripotent stem cells. The induced pluripotent stem cells are the adult cells that have genetically reprogrammed to an embryonic stem cell like state by being forced to express the genes and factors, which is very important for maintaining the defining properties of the embryo stem cells. Next, I'm coming to the important characteristics of pluripotent induced cells are induced pluripotent stem cells. These cells made the defining criteria for pluripotent cells. It's not known if the induced pluripotent cells and the embryonic stem cells differs the clinically following the significant ways. The human induced pluripotent stem cells also expresses the stem cell markers and are capable of generating the cells and characteristics of all 3 germ layers. What are the all 3 germ layers? Ectoderm, endoderm, mesoderm. Very simple. Right. Next, in continuation of the induced pluripotent stem cells. Although the additional research is basically needed for IPS. The induced pluripotent stem cells are already in use for the drug development and the modeling of genetic disorders, and the scientists hope to use them in the transplantation medicine. Right. And here I would like to tell you the important point. The stem cells are the greater alternative to the transplantation procedures. So in the transplantation procedure, always there are 2 problems, donor, recipient. So what is the donor recipient problem? The cells should be matched, blood group should be matched, organ should be matched, and even though 30% only the success rate of the patient. So it may be a kidney transplantation, it may be liver transplantation, it may be pancreatic transplantation, whatever it may be, it's a painful procedure. Transplantation is a very painful procedure, very difficult to bear the pain of transplantation. So stem cell based therapies is the only alternative to the transplantation. Very simple. A single cell obtained from the patient, it will be introduced into appropriate cell culture medium. The cells are cultured, the cells are harvested, and the cells are transplanted directly to the patient in the appropriate infected part. And what happens? Very simple. The healthier, younger cells are able to destroy the diseased or dysfunctioned cells. Slowly, the transplanted cells can give rise to the complete organ structure. It slowly functions. So there is no need to transplant any organ or any tissues from one human to another human. Very simple, only one cell is enough to make all those things. So, and next I'm coming to, the viruses are currently introduced the reprogramming factors into the adult cells. And this process must be carefully controlled and tested before the technique can lead to the useful and treatment for the human being. It's a very, very dangerous experiment. The reprogramming of virus. What's the example? Coronavirus. The reprogrammed virus. The genetic system of virus may be reprogrammed. So non-pathogenic to highly pathogenic in nature. Next I'm coming to in continuation of induced pluripotent stem cells. The researchers, that is scientists, are currently investigating the non-viral delivery strategy. Non-viral delivery strategy, underline. In any case, this type of breakthrough, the discovery has created a powerful new way to differentiation of the cells whose developmental fate had been previously assumed to be determined. Right. Next, I'm coming to in continuation to the induced pluripotent stem cells. In addition, the tissues are derived from induced pluripotent stem cells will be nearly identical and to the cell donor. So therefore, probably avoiding the immunological rejections by the immune system. Sometimes what happened, our own immune system destroying our own tissues or our own cells. That is called autoimmunity. What is autoimmunity? Why the autoimmunity is so vital for the transplantation procedure? Any material or any foreign material which is entering into the human body, it will be considered as antigen. The immune system. Sometimes by mistake, the immune system wrongly calculated. Our own system, our own immune system is misunderstanding and it consider our own organs as the foreign. So the immune system trying to attack our own cells, our own tissues. This is the autoimmunity. Right. Next, I'm coming to the induced pluripotent stem cell strategy creates the pluripotent stem cells that together with studies of other types of the pluripotent stem cells which help and scientists learn how to reprogram the cells to repair the damaged tissues in the human body. Next, I'm coming to what are the sources of the stem cells? What are the sources of stem cells? There are many sources of the stem cells. I'll give you one or two points regarding the sources. Basically, the stem cells may be derived from the autologous, allogenic, or xenogenic in nature. Histocompatibility is prerequisite for the transplantation of the allogenic stem cells. The fetal tissues is the best current tissue source for the human neural stem cells. However, the ethical issues are a major concern. Next, I'm coming to the potential sources of the stem cells. What are the potential sources of the stem cells? They are as follows. The fetal tissues that become available after an abortion exists. The embryos from assisted reproductive technologies, that is ART, assisted reproductive technology, such as commonly used in the fertility clinics. And the embryos created through the in vitro fertilization process are specifically for the research purpose. This is prohibited for many nations. Right. Next, I'm coming to the sources of stem cells in continuation. So the embryos created asexually as the result of the transfer of a human cell nucleus to an egg with its own nucleus removed. The other sources of the stem cells are those from the umbilical cord blood and the bone marrow. In addition, the neural stem cells, hematopoietic stem cells, and the mesenchymal stem cells can be harvested to form the fetal blood and fetal tissues. Next, I'm coming to the placenta, a source of stem cells. The placental stem cells, like umbilical cord blood and bone marrow cells, can be used to cure the chronic blood-related disorders such as the thalassemia, leukemia, and sickle cell anemia, etc. Next, the umbilical cord blood system and the cell transplantation. Basically, the umbilical cord blood stem cells, the transplants are less prone than either bone marrow or the peripheral blood stem cells. This is probably because the cells have not yet developed the features that can be recognized and attacked by the recipient's immune system. I told you, just remember autoimmune system. Next, I'm coming to the applications of stem cells. There are a lot of applications of the stem cell. In the laboratory, the scientists can follow the stem cells as they differentiate themselves, and it becomes increasingly specialized, making the skin and bones, brain, and other cell types, and identifying the signals and the mechanisms that determine whether a stem cell chooses to carry on the replicating itself or differentiate into the specific cell type, and into which cell type will help us to understand what controls and normals development. The next point of applications of the stem cells. The stem cells may help us to understand how a complex organization from a fertilized egg, a better understanding of genetic and molecular controls of these process may yield the information about how such diseases arise and suggest the new strategies for therapies. This is an important goal of the stem cell research. Next, applications of the stem cell. In many cases, it is difficult to obtain the cells that are damaged or diseased and to study them in the daytime. Next, the stem cells, either carrying the disease gene or the engineered to contain the disease genes, offer a viable alternative. So the scientists could use the stem cells to the model disease process in the clinical laboratories and better understand what goes wrong. Next, I'm coming to the stem cells could provide the resource for testing new medical treatments. Yes, The new medications could be tested for safety on the specialized cells generated in large numbers from the stem cell lines. It's reducing the needed for animal testing. Remember, I told you. So, stem cell biology or the stem cell engineering or regenerative medicine is the greater alternative to transplantation as well as the animal model experimentation. Here there is no need of animal for usage. So the new medications could be tested for the safety and the specialized cells in the generated large numbers from the stem cell lines, reducing the needed for animal testing. So the other kind of cell lines in ready to use industry. Cancer cells or cancer cell lines, we can say a great example are used to screen the potentiality of the anti-cancer agents or anti-cancer drugs. Next, I'm coming to cell-based therapy or cell therapy. So the treatment of neural diseases like Parkinson's disease, Huntington diseases, and Alzheimer's disease, the stem cells could be used to repair or replace the damaged neurons. So the repair of damaged organs such as liver, kidney, pancreas. So even though we are treating HIV also. Next, the important question arises. How does the cell therapy works? What is the basic mechanism behind the cell therapy? So the stem cells can be used to generate the healthier and functional specialized cells which can then replace diseased or the dysfunctioned cells. It is similar to the process of organ transplantation, only the treatment consists of the